All right. Hello, everyone. My name is Dr. Tuan Chow. I'm a child neurologist and physician scientist at the Jan and Dan Duncan Neurological Research Institute, which is affiliated with Texas Children's Hospital and Baylor College of Medicine in Houston, Texas. And today I'm going to share with you a presentation that was given at the 2019 EBF3 HADDS Syndrome International Family Conference that was held for the first time in Houston, Texas. And the story today is about the discovery of HADDS syndrome. So what is a neurodevelopmental condition? This is an understanding that we need to establish at the beginning to understand this story. A neurodevelopmental condition results from alterations to the growth and development of the nervous system, and it can have wide-ranging effects on emotion, cognition, and behavior. Typically, the symptom onset or recognition of this condition occurs early, at less than 3 years of age, due to delays in achieving important developmental milestones. However, the severity can vary widely depending on the underlying cause. And this is of importance to all of us here because globally, 7 to 8 million children are born each year with some type of a neurodevelopmental condition, but only 30 to 40% have an identifiable cause. So the challenge that we all face is really trying to understand what is happening for the other 60 to 70% of children that affects how their brain is developing and how it's working to lead to the different challenges that people have to face with a neurodevelopmental condition. So, extensive research efforts and the support of many families like yours has opened our understanding of many, many genes that are important for how our brain forms, how our brain is able to function and do many of these complex tasks and capabilities. But there's still a gap in understanding the full picture of what is contributing to brain development and function. And why is it that when they go awry, we deal with these different complications in terms of how our brain is able to function and process information. And one such gene that I'll share with you today is called EBF3, or early B cell factor 3. Now, the story of EBF3 and what we've come to understand about the human neurodevelopmental condition really begins for us here in Houston at Texas Children's Hospital in October of 2015, when I had the immense privilege of meeting a very special young girl and her family who came with a story very similar to many of yours, with delays in achieving motor and language milestones, but otherwise was quite healthy. And all traditional testing that we were able to do clinically really wasn't giving us a direct answer. And this mystery really began to intersect with the story of EBF3. What we did not realize at the time when we first met this young child in 2015 was that shortly thereafter, in the spring of 2016, I was beginning to study a very interesting gene called EBF3 as part of a large-scale research effort conducted with the Undiagnosed Diseases Network and the Bellin Laboratory at Baylor College of Medicine and the NRI. So what is the Undiagnosed Diseases Network? So this is a nationwide effort across multiple sites that actually was initiated initially at the National Institutes of Health and then expanded to encompass the entire nation, where individuals and their families with rare conditions which have eluded traditional medical diagnoses can self-refer to this network and participate in research studies that hopefully will lead to much needed answers and at least a direction to begin to understand their diagnostic challenges. So once individuals refer themselves or their physicians can help refer them to the UDN, they are then assigned to clinical sites closest to where they reside. Once they're part of the UDN team, they undergo a very comprehensive evaluation of all of their medical issues and concerns. Then their genes are sequenced in search of potential genetic changes that may be impacting how that gene works in such a way that it could explain the different challenges they are facing. Depending on the type of gene affected, the model organism screening center team will step in. They will look at those genetic changes and try to understand which of those is a likely candidate to be studied in different animal models, such as the fruit fly, the zebrafish, or the C. elegans worm. And once they are assigned to one of these teams, then efforts begin in earnest to begin to understand how those changes are contributing to the human features. So our story with the Undiagnosed Diseases Network that began early in 2016 is around a young boy who had a history of low muscle tone and delayed development when walking and talking. He dealt with challenges in the ability to coordinate his speech, maintain his balance, help with his swallowing. And he had some interesting features like decreased perception of pain, decreased strength in facial expressions, and he had a history of strabismus that was surgically corrected. Now, almost 7 years prior to us beginning to work on this gene at Baylor College of Medicine and the NRI with the UDN colleagues, he and his wonderful family were enrolled with the Undiagnosed Diseases Project, the predecessor of the UDN, at the National Institutes of Health for the study and diagnosis, hopefully, of a rare condition. At the time, there was a genetic test called a whole exome sequencing that was only available on a research basis. It was not yet available clinically. And so whole exome sequencing was conducted for him, and the NIH team identified that he had a de novo, meaning not inherited, it arose anew in just him, genetic change that was of unclear significance, meaning unclear impact on how the gene works, in early B cell factor 3 or EBF3. And this caused the amino acid arginine in position 163 to change to a glutamine. So this boy did have some findings on neuroimaging that suggested that there were some effects on the development of the cerebellum. So here you can see the cerebellum circled in yellow, and parts of the cerebellum in the middle, as indicated by the arrowhead, or on the side, structures as indicated by the asterisk, all showed indications that there was some development that was altered in this very important part of the brain that plays a role in motor control, and it plays a role in some other non-motor functions as well, such as speech and language. So, but this was just one child, and it really, is hard to understand the impact of single individuals' gene changes because all of us have so many of them. It's part of what makes us unique. And to understand better if this gene being altered could be contributing to a neurodevelopmental condition, we needed to try to see if there were other individuals who were also similarly affected. So we quickly reached out to our colleagues at the Baylor Genetics Diagnostic Laboratory, and they curated their data sets to see if there were other individuals who also had rare misspellings in this gene. And indeed, we rapidly found 2 other individuals, all had whole exome sequencing, and they all had found a misspelling in the early B cell factor 3 gene that was not inherited from their parents. So very similar to that young boy. Both were girls. One was based at Texas Children's Hospital, the other one at NYU Langone. Both children had very similar features, and one of them had underdevelopment of parts of the cerebellum that was similar to what we saw in the young boy as well. And they all had misspellings in the EBF3 gene affecting the exact same amino acid, this arginine 163. So what is EBF3? So EBF3 regulates expression of many genes, and so we consider it as a master controller. Think of it as like the architect for building a particular structure. And it needs to know and function properly to turn on and off certain genes at the right time and right location. It is present and nearly identical from fruit flies to humans, which tells us that this gene probably plays a very important role in development, in such that change in this gene is undesired. So what was happening with this EBF3 changes? So through a series of studies that we conducted in the research laboratory, what we found was that loss of one functional copy of the gene, either through misspellings that just changed one portion of the gene or a deletion of a whole copy of the gene, causes a reduction in its ability to regulate gene expression. So if it can't be a master controller working at full capacity, it's going to have difficulties in making sure that the complexity underlying how our brain develops and functions is affected. Now, EBF3 and all the other members that do similar functions are present across nearly all species. So whether it's the humble worm, fruit fly, up to fish and frogs and mice and humans, it's present and it's very, very similar to each other. There's very little change that has happened over time. And so we were able to utilize this approach to really begin to ask the next question, which is these effects of these mutations on how EBF3 is regulating gene expression. Could it be playing a role in a more complex development of a living organism? Is it truly contributing to impacts on how the development is supposed to occur? So we turn to the fruit fly because in the fruit fly, there's only one member of this factor. There's one version of it. And in the fly gene, it's called NOT, also known as Collier. And it was named because over 50 years ago, when this gene was first identified in the fruit fly, some of the misspellings would affect how the fly wing formed. It looked like a knotted wing. So what is known in the fly is that this gene does play a very important role in regulating brain and nervous system development. So here I'm showing you an image of a fruit fly embryo, and you can see in black the areas that NOT, the gene, is expressed in, and it's very high in the nervous system. So in red, I've circled here the developing brain, and the other structures here are analogous to like spinal cord and other neurons and nerve cells in our body. What's known in the fly literature is that this gene is highly critical and essential for normal development and survival. So you really need this gene to be there and working at max capacity to go from a little embryo to a healthy adult fly. Completely losing both copies of this gene is detrimental and it prevents development from the embryo stage. So it creates a lethality or stops development. So we wanted to ask the question of, can we use the human EBF3 gene, the typical version, to functionally replace the fly gene? Because if that's the case, if the human gene can work in the fly and function appropriately in regulating development all the way from embryo to an adult, then we could potentially study the effects of these human alterations directly in the fruit fly in a developing animal and identify if it is having an effect on very complex processes. And indeed, that is what we found. We were able to show that the typical copy of EBF3 that we see in most individuals is able to work just as well as the fly gene, not in the fruit fly. And it can go from an embryo all the way to a healthy adult. But when we put in EBF3s that carry the misspellings, the mutations that we found in those 3 children, they couldn't work appropriately. The EBF3 that was misspelled halted development early in the embryonic stage, which tells us that those misspellings, even though they affected just one amino acid out of the whole protein, was sufficient to prevent EBF3 from functioning appropriately. And so this proved definitively that those misspellings were detrimental. The findings from our team with the Bellin Laboratory and Baylor College of Medicine's UDN team came together with 2 other international research groups for the first recognition of EBF3 and its relationship to a human neurodevelopmental condition. And these studies were published online in December of 2016 at the American Journal of Human Genetics. This was subsequently available in hard copy format early January of 2017. And together, the 3 studies, which I show you here, represented a total of 21 patients. 21 families participated in these research endeavors and led the way to understanding solving at least a question of what is the gene underlying the challenges they're facing. And these 21 families has opened the door now for many other people to have much needed answers, and for us to now begin to understand better this condition and better what might be able to be done for this. So I have here our research team and other colleagues at Baylor College of Medicine underlined in red, which contributed to 2 of these 3 initial discovery studies. Subsequently, in 2017, 3 other studies were published, representing a total of 7 additional individuals that all were identifying that there were rare mutations in specific parts of the EBF3 gene, or a whole gene deletion of just EBF3 that all shared the same features. And continued understanding of this condition is now further improving because of the support of families like yours and their participation in these research efforts. So together, these first reported EBF3 gene mutations in 2017 begin to tell us what parts of the gene and what parts of the protein that the gene encodes is affected. And for most individuals, these misspellings are affecting a part of the gene that helps this gene, EBF3 protein, bind to DNA, and therefore the binding to DNA is needed for it to function and regulate gene expression. Now, together, all of these efforts led to the recognition of a condition now known as hypotonia for low muscle tone, ataxia for the gait imbalance, difficulties with coordination, and delayed development, or HADD syndrome. And this condition encompasses many of the features that you've just heard about, as well as a risk of some febrile seizures, of difficulties with motor control, of difficulties with facial expressions, effects on their language development. And these children and individuals, some of them will have disturbances that can be seen in the cerebellum structure. Some of them will have effects on their urinary system, their bladder control. And together, these are all tied to these misspellings in EBF3. So to better understand this condition, it was so recently identified, there was very little information known about it. We realized that we needed to work and understand this from the perspective of the parents of the individuals who are affected. Because out of everyone, out of all the research teams, it is you, it is the families who understand this the best, who are the experts in this condition and what is of impact and of importance to study further. So in 2017, in that summer, myself and our colleagues established a concerted clinic for EBF3 related HADDS syndrome or other EBF3 gene deletion syndrome clinic, so that we can understand this condition better, that we can provide clinical expertise for a rare condition, and then understand also from the families what is happening that is affecting the quality of life. And then in October of 2018, these efforts were then formalized to an EBF3 clinical study, which is based with my research team at the Jan and Dan Duncan Neurological Research Institute. So I want to thank all of you, because many of you here have supported and have contributed to these research efforts. And it truly has given us great strides forward in better understanding what it means to have these misspellings in EBF3, what is the future implications of this condition. And we hope that this will lay the foundation for us to begin to understand what could be potentially done to improve the quality of life for everyone. So the next step in the EBF3, HADD syndrome, and other related deletion syndrome research efforts is to, one, advance the understanding of this condition. We really need to understand what it means to be a child with this condition, what it means to be an adult with this condition, what it is that we can anticipate and plan for, and really begin to understand how it is affecting the brain, both in a developing stage and also when it's mature and it needs to function for many complex tasks. We need to begin to understand the medical history, all of the complex issues. We talk a lot about the brain and what EBF3 is doing there, but we also know that individuals with these conditions have medical issues spanning not just the brain, but it affects their bladders, their kidneys, it affects their GI tract, many, many parts. So we need to understand what it is that's the whole constellation of this condition, because it begins to tell us also where we need to look to study EBF3 better, to understand what is going on and what can be done to minimize those complications. We want to understand the diagnostic studies and the results of that. And we need to understand, how does the gene mutation, misspellings, relate to these symptoms? How does it affect neurocognitive behaviors? How does it affect how you interact with the world? How does it affect how the brain functions? So these are different avenues that we are now pursuing through our clinical research efforts with all of your support and assistance. The mirroring half of that is to take those understandings and bridge it with what we're doing in the laboratory at the NRI. And so here, my research team and those of our collaborators are really beginning to advance our understanding of fundamental biology. What is this gene doing? What is it happening in the developing brain, in the mature brain? What is its specific role in how our brain is supposed to form and function? And we're doing this by understanding not just the parts of the brain involved, but also the different genetic pathways, the different genes that are dependent on EBF3 to appropriately turn on and turn off as our brain matures and functions. And so all of this efforts, all the knowledge that we've gained so far, really is driven by you all. You are our inspiration. You are without a doubt our heroes. Several of you have been able to come and visit our research team at the clinic, visit our research laboratory. And you know that many of you all, we see you all every day. Your smiles, your hopes, your support, it is a place of honor in our laboratory. And we keep you always in mind. And all of this efforts, both those who have contributed to our research studies, to those of our collaborators and other colleagues in the field, thank you very much. You have been the driver behind many of these discoveries, and you continue to be in so many ways. And so your expertise is absolutely invaluable. So thank you. Thank you to all of you for inspiring us. And we look forward to working with you to finding many important answers. Thank you so much. Bye-bye.